Hi, Michelle Glass here. Welcome back to our next installment in our Chapter 3, Lecture 3 series. And our topic for this video is how proteins are made. So we're going to be looking at this key process. Let's just take a minute and remind ourselves of the significance of those proteins. So what are the jobs of the proteins in the cell? We've talked about them as enzymes, which are essentially controlling chemical reactions. So cells can regulate what chemical reactions they can perform based on what enzymes are available. We talked about proteins being involved in self-recognition and our immune response, structure to both cells and tissues, and so the body is composed of cells and tissues, so this is essentially the structure of the body. Hormones and communication is an important job here. Cell membrane channels, which is another way that proteins are involved in communication. So we're seeing that proteins have significant jobs in our cells and in our bodies. So let's look at this process. So the basic steps of making a protein starts with a process called transcription. And this is occurring in the nucleus. So we will see that the cell uses the DNA molecule in order to construct an mRNA molecule, where that little m is standing for messenger. So in the nucleus, this is where the DNA is always located and housed. We see the process of transcription taking our master instructions and then just reading off a little bit of that information for that protein and turning it into a messenger RNA. And then the job of the messenger RNA is to take that information to the ribosome so that protein can be made. Remember, our ribosome is our location of protein synthesis, and so that's why the mRNA is going to take that information out to the ribosome. This process, the second basic step, is called translation. So here, if you want to use your alphabet, you can remember that C comes before L, so transcription comes before translation. So here is our overview of how a cell makes a protein. It starts with that DNA molecule, which is our master code. Because DNA is always stuck in the nucleus, and that process of converting our master code into maybe like the little recipe card is called transcription. The mRNA is just that gene, the information necessary to make that one protein that the cell wants to make. That mRNA is small enough to leave the nucleus, and it takes that information to the ribosome where translation happens. That's the process whereby that mRNA code gets converted into a protein sequence. Okay, so let's revisit our DNA molecule just a minute. So DNA is, of course, standing for deoxyribose nucleic acid. Often you can talk about the basic structure as looking like a ladder. So it's a double strand. So you have two sugar phosphate backbones, and then the rungs of your ladder are your nitrogenous pairs. Remember, our nitrogenous pairs can be either adenine, which pairs with thymine, or cytosine, which pairs with guanine. And remember, we have that rules of complementary base pairing that I already introduced to you when we discussed nucleic acids. Remember, too, that thymine is only found in the DNA molecule, so we won't see that when we start looking at RNA structure. The DNA molecule has the instructions for that primary protein sequence, which is essentially what is the order of the amino acids when we're building our protein. And the DNA molecule, remember, is trapped, it's stuck in the nucleus. So it can't take those instructions directly to the ribosome. And that's good because this is your master copy, right? You don't want anything to happen to this. So maybe this is like grandma's recipe book and she never lets it leave her kitchen. And so if you want a copy of her recipe, you have to come in and write it down on a note card and maybe take a photo of it with your phone so that you can have that recipe actually leave her kitchen. So you might want to think about it in that way. mRNA is that photocopy of the recipe. It can copy that gene sequence and take the message to the ribosome. Now, a gene is the instructions for how to make a single protein. And so, excuse me, I got click happy. And so when we're talking about making this mRNA, we're talking about just copying the information from a single gene in order to make one protein over at the ribosome. Not just like a single protein molecule, we're going to make it repeatedly. So we're going to make a lot of that one type of protein for the cell. Okay, so we are ready to here look at our basic steps. We're starting with our DNA molecule. We're inside the nucleus. We're doing the process of transcription, which means to convert the DNA sequence into an mRNA sequence. So that's what we're ready for here. So put my picture down there. All right. So here I've kind of simplified DNA structure, right? Notice I have my complementary base pairing across. I have adenine pairing with thymine, and I have guanine pairing with cytosine. We're inside our nucleus here, which is bordered with a nuclear envelope. There are pores in the nuclear envelope, and DNA is too big to fit through those pores. That's why it's trapped. But the mRNA is going to be able to use those pores to actually exit the nucleus. Our mRNA is, again, standing for messenger ribose nucleic acid. Recall there's a few differences in structure. It's single-stranded. It's composed of the ribose sugar instead of deoxyribose. It has uracil as a base instead of thymine, but the rules of complementary base pairing still apply. So here, adenine will pair with uracil, and cytosine will pair with guanine. Okay, so if we're going to read off that top DNA sequence there and build our mRNA, we're just going to use our rules of complementary base pairing. So where we see a thymine, we're going to put in an adenine. Where we see an adenine in DNA, we're going to put in a uracil. And where we see a cytosine in DNA, we're going to put in a guanine. So when we see a sequence of TAC in the DNA, it's going to pair up with an AUG sequence in mRNA. So see if you can do that next set of 3. Okay, so let's see how you did on that next sequence. All right, so where in DNA you had a GCA, you should have put in a CGU for your mRNA sequence. And see if you can finish it up. So taking a look at that rest of the DNA strand there, you have a GGC. So what would pair with that in the mRNA? Okay, good, CCG. Excellent. So this is the basic process of transcription, where you see the cell is reading off of the DNA molecule using the rules of complementary base pairing. Lots of specialized enzymes are involved to create this mRNA strand. So now we have a copy of the code that's able to leave the nucleus. Now what we see is that the mRNA is actually going to be edited before it leaves the nucleus. We see that parts of the RNA are cut out and basically thrown away, and then other things are pasted together. So you have like some cutting and pasting, and that allows us to take one sequence and modify it to make different proteins. So sometimes you hear it described as like, you know, using different punctuation. So you can have the same words. Let's eat, Grandpa. And then you could say, let's eat, Grandpa. So based on the differences in your speech pattern or your punctuation, you have 2 different meanings in the same sets of words. So we can kind of think about our editing as something like that. This edited mRNA leaves the nucleus and goes out to the ribosome, which is of course where our next step is happening. So when we're talking about our next step, we're looking at translation, which is the process of taking an mRNA sequence and at the ribosome converting it into that amino acid sequence, which is our protein. To help us at the ribosome, what we see is that our mRNA strand is coming in, and then you have this tRNA molecule. The T stands for transfer RNA. The tRNA molecule, I like to think of it as having like hands and feet, and it's holding on to an amino acid with its hands, and then it's holding on with its feet to what's called an anticodon. So before we define the term anticodon, let's look at our word codon. So let me get my pointer here. I have my word right here. So codon is talking about 3 bases together that code for a single amino acid. So 3 bases coding for 1 amino acid is our codon. So our first codon here is AUG. Our second codon is CGU, and our third codon is CCG. So you can describe this as a triplet code because you're reading it in groupings of 3. An anticodon is opposite to the codon. So if the codon is AUG, the anticodon is UAC. We're just using our rules of complementary base pairing. tRNA is RNA, so that means that it's going to have uracil instead of thymine. So the job of that tRNA molecule is to bring in those amino acids, and it locks into place based on the recognition of the anticodon for the codon, and that's how we know the order of those amino acids to bring into place. The other thing to note here is your ribosome is composed of our third type of RNA, which is the ribosomal RNA, our lowercase r, big R in A, okay? So what we see happening here is that this tRNA is going to bring in our first amino acid based on the recognition with the anticodon. The mRNA is going to shift so that we can get a second tRNA coming in, locking into place with our next amino acid. When you get 2 amino acids close together, they're going to form a peptide bond. And so we're starting our structure of our protein. So this is what the cell is using. For humans, we can figure out the amino acid sequence based on what's described as a codon chart. So it's already been determined for us that this codon codes for this particular amino acid, and that's what we're seeing here. So we're going to be using our RNA codon expression table, and we know that this is RNA because we see uracils. Up above, we have thymine, so that tells us that this is DNA. And so what we want to do for that first codon, we had an AUG. So we look for our AUG, and then on the other side, we get our code for our amino acid, which in this case is MET. So that is our first amino acid that we're going to put into place. So that first amino acid that was brought in was, it's methionine. You don't have to know that, but that is what the MET stands for. So now let's see if we can figure out what is that second amino acid. So we have a CGU. So we want to look over here for CGU. And we can find our next amino acid is abbreviated ARG, or I think that's arginine. And then here we're seeing the formation of our peptide bond when we have these 2 amino acids in close proximity. We could even look for the CCG if we wanted. So if we did that, I found it here. That third amino acid would be PRO. So that stands for proline. Okay. So that's really what we need to be able to do for transcription. Okay, so again, here's our overview. We see transcription. I'm not sure if I said the right word a minute ago. Transcription happens first, translation happens second. Transcription is occurring at the nucleus. Transcription is DNA being converted into an mRNA sequence. And then translation is the process that happens at the ribosome, and this is where you take that mRNA sequence and use it to make your protein. And that's it for now.